today about the work we did at ID2 about the assembly mechanism of viruses. And in general, okay, so the challenges we are trying to address in our lab include, there are three challenges. We try to resolve the structures of very large biomolecular complexes that are forming in solution. And then we try to determine the underlying physical interactions that lead to these assemblies. And then the dynamics of self-assembled structures that can either attain equilibrium or operate out of equilibrium. Today I'll focus on structures that attain equilibrium, these are the viruses. And icosahedral viruses are about half of the known viruses. And the icosahedral viruses have the icosahedral shape of various sizes. They are called, they're giving the T number correspond more or less to the number of subunits that make the capsid, the envelope that envelop the nucleic acid. And the capsid protein is the shell. And in the life cycle of a virus, the virus enters the cell. It has to disassemble, release its genetic material into the cell. The cell is using the machinery of the cell to make copies of its components, the proteins, and the genome. And eventually there is, in the life cycle, the assembly. They have to assemble together and exit several copies of the same virus, have to assemble in the cell and eventually get out of the cell. So this is the assembly phase where we will focus today on the talk. And on the one hand, the product has to be stable enough to survive all these processes outside the cell and inside the cell. But on the other hand, it has to disassemble. So there has to be some, it cannot be too stable because otherwise there will be no way to disassemble it. So, so we, in this talk, I'll talk about hepatitis B. And it turns that 90% of the hepatitis B virus, hepatitis B has is infecting actually a third of the population. The world population has been infected by hepatitis B, but only much smaller fraction, only about 250 millions a year. It's $250 million. Suffer from it, from chronic disease. Although there is a vaccine for it, they still suffer from the chronic disease, and about 800,000 people die from it every year. It is a relevant virus in many aspects, of course, but it's also a very interesting virus and a very well-studied virus. It turns that you can assemble the virus-like particles in vitro. And in the cells, 90% of the hepatitis B virus remain empty, either as a Trojan horse or as a storage form or as immature particle for subsequent packaging of the genetic material. So and there are other reasons, from many other reasons, people are studying virus-like particles, also as protein nanocapsules for drug delivery application, as nanoreactors for catalysis, as a scaffold to display antigens for vaccine applications, or as sophisticated higher dimensional materials, functional materials, biomaterials. So in all of these cases, there is an interest in both the structure, the stability, and the dynamics of the assembly process. So the goal of our study was to study how the empty capsid of hepatitis B, which is well-behaved, well-characterized virus, there has been, it's in collaboration with Adam Zlotnick from Indiana University, who studied this virus for years. but the assembly pathways to start from the subunits, the protein subunits, the protein capsid subunits, they are dimeric, and they go all the way to the final capsid, which can have 120 copies of the same dimers, that then it's the T equal 4 symmetry, icosahedral symmetry with a diameter of 33 nanometer, or the T equal 3, which contains only 90 dimers and a diameter of 28 nanometers. The majority is the T equal 4 particles, but they exist also in vitro and in vivo. You can find also the smaller particles. And this pathway can be super complex, because it turns that from our enumeration of the states, it turns that there could be 10 to the 30 possible intermediates on the path from the dimers to the capsid. So this generates a huge complexity. And then if you think about the assembly pathways, there could be many more possible ways to assemble the capsid. So apparently this is an ill-defined problem, but it turns that we could address it. So the interesting thing is that this interaction involves relatively weak protein-protein interactions. And the idea was really to find what is going on in the middle, what are the rates, what is going on really, what is the mechanism and the pathway for starting from the dimer all the way to the capsid. So we use Solution X-ray scattering, where we focus the X-ray beam into where in this audience you probably know what it is. You focus the beam, you scatter from the virus in the solutions, and then there is the two-area detector where data in scattering intensity is a function of scattering vectors or scattering angles are generated after the integration. And here the analysis is really the part that is challenging. We could get temporal resolution of milliseconds, and it's, of course, it's label free, and so it has all the capacities that we needed. However, data analysis, as I said, is really the challenging. So one of the things that we did in the group, we developed analysis softwares over the years. We developed X-ray scattering. They are available in our website, where the first software was developed 10 years ago, more or less, where we modeled geometric subunits with multiple shells, like sphericals with multi-shells, with radial electron density profiles that can be made of many shells and so any profile could be studied. And then we did it for other geometries like cylindrical, helical, or layered structures, but this was not enough in this case. So 2 years ago, we published a much more advanced software for data analysis of solution scattering. It's called DPLUS, where we could dock subunits in a hierarchical manner and describe the assembly symmetry of each subunit. So the dimer, for example, can be docked into the icosahedral assembly symmetry of the virus, the capsid. And in that way, we essentially can model, any structure, because we can do it in a hierarchical and with multiple subunits. And we could use either atomic models, or if they are available from the PDB, or geometric models in case they are not. So in that way, we can model what should be the scattering curves of very complicated structures. So this is one challenge that was on the way. And just to demonstrate what it looks like, if you see the scattering curve, this is the intensity as a function of the scattering angle from the dimer. The blue symbols are the model, the gray bars are the noise, or the error bars of the data. And the red curve is the atomic model of the dimer taken from the protein data bank. And the red curve is the model that is expected from such a dimer in the solution, where you have all the possible orientations, and therefore you have to take it into account. The other extreme is the case where we have the full capsid, and these are the data, the blue, and you can see that this is an atomic model where we take the dimer, put it in the assembly symmetry, the icosahedral symmetry, and we just need to fit the radius or the diameter, and as you can see, the fit is really, really good. Okay, so now what we did, we studied first we studied the phase diagram, the conditions where assembly can take place, and the conditions where assembly does not take place, and we were wanting to know in each case at equilibrium, what are the components in the solution that can explain the data. So as you see here, different 3 temperatures, 9 degrees, 25 degrees, and 36 degrees. This is the temperature axis. And here you see the curves at different salt concentrations, ionic strength. So as you increase, as you can see here, the scattering from the dimer looks similar to what we have at low salt. And as we increase the salt, this is we start to see the oscillation that corresponds to the formation of the capsid. But we don't have only the capsid, as you can see here. It's not exactly what I showed you when we have a lot of capsid. We have also coexisting structures in the solution that take place. So in order to know what are all of these structures that can explain this data, what we did is what ESO did here, the first author, he did simulation, because now, as I said, there are 10 to the 30 distinguished intermediates. That's also the number. This number is also coming from Roi's simulations. He did umbrella sampling of Monte Carlo simulations, where he sampled and described the possible states, but of course not 10 to the 30. It's impossible. So we had to do is we had to in the phase space of degree of connectivity, which means the number of contacts between the subunits, and one correspond to a compact structure with the maximum number of contacts. 0 correspond to the most open structure with minimal number of contacts. And then here, the second axis, this is the vertical axis. The horizontal axis is the number of dimers. So we start here from a dimer. Here we have the full capsid. So this is one state here. There is only one state. But in between, as you can see, the heat map here shows you that in the middle here, you have a lot of states. You have 10 to the 28 states where it's in the intermediates of about 80 dimers. This is highly populated. There are many, many options. And the heat map shows you the density of states that can be in all of these, for all of these intermediates. So this is a situation where it's impossible, because from entropic reason, it's a highly, most of the entropy is located here, but these states, the open states, are less stable. So in order to really, so holy capsids are here, and they are favorable by entropy because there are many, many states here. And to explain the data, we had to take into account the stability. Okay, so what we did here is we used this enumeration of states as to explain the data by modeling representative states, only a few thousand. You don't need all the 10 to the 30. It's impossible even to calculate so much. So we had about a few thousand representative states of the intermediates in the solution. And what we did is, by employing a thermodynamic model of self-assembly, we computed the molar fraction of each state by taking into account the dimer-dimer association standard association free energy. This is the delta F here. And the density of state, this is the omega, representing the number of states that correspond to a given same number of dimers in each state, and the same number of contacts. So we grouped them into families because they had similar stability of overall thermodynamic stability. And then we balanced it with the entropy of the dimer being in the solution. So imagine you can either have all the dimers separated in the solution, gain entropy from it, or assemble and gain the association energy between them. And then you have also the entropy of the degeneracy of the number of states that you can, of similar stability. So this is based on the thermodynamic model. We could get the weight of each, the molar fraction of each model intermediate. And this is what explains all of these data. So all of these data were based on the fitting only the standard free energy of association between the dimers. And by doing this, we could explain the entire phase diagram of temperature as a function of salt. All of these were modeled very nicely in using this thermodynamic bias. So we could filter out the most important states that contribute to the assembly in each of the conditions of the assembly. So what we found is that 99% of the mass was found either in dimers or in full capsid, either T equal 3 and T equal 4. And the rest, most of the rest of the mass was on the 1%, was maybe 2 and 3 dimers, and the rest was really a negligible amount. As we increase the salt at each temperature, as we increase the salt, we see that the fraction of the dimers decreased, and the fraction of capsid in T equal 4 and T equal 3 started to increase. The same happens at the higher temperatures, but at higher temperatures, less salt was required in order to form the capsid. So this was one thing that we did and was explaining the data very well. And the results showed a very interesting effect. We found that no assembly occurs when the association energy was in thermal energy units, only 7 kT. Below 7 or 7.5, there was no assembly. Then between 7.5 and 8.5, so about only 1 kT difference, there was successful assembly. When we went to higher association energies by going to a higher salt, so this is the Bias-Kruning length, which is one inverse of the salt. So this is a high salt part where you see the association energy is strong. And so here there was no assembly. So successful assembly was only in a narrow window. You can also see it in the EM here. And when we went to too much salt, we started to get malformed particles, as you can see here in the EM. So assembly was not successful. So there is a very narrow, so to answer the first question, how do the viruses manage to go through all the life cycle that requires assembly and disassembly? It means that there is a very narrow window of association energies that lead to successful assemblies. And that's how so only 2 kcal per mole difference in the standard free energy for association between the dimers can lead already to either formation or no formation, is the difference between no assembly and malformed particles. So there is a very delicate balance. So this gives hope that actually drugs can be generated in order to tune this association energy, small molecules or maybe even osmolytes can do it. So this is the case in equilibrium. And then the pathway for assembly was resolved by time-resolved SAXS using the stop flow setup at ID2. And where we, in one, we had on one hand the dimers at low temperature, and the second injection was the salt solution that was enabling the assembly. So by mixing the protein with salt, we could generate the assembly. And here we kept the temperature at the higher temperature. And then we took data that looks like this. This is the time-resolved data. As you can see, as the time goes by, you can see an increase in the depth of oscillations that generated the data, which means that there is assembly. Now, in order to resolve now the pathway for assembly, we used another method, another algorithm to analyze. These are now the data, and you can see here two sets of data, one in mild assembly conditions, when the salt concentration was close to physiological. And here there is a high salt concentration which correspond to aggressive conditions that lead to more viruses, but also the pathway is very different. And the timescales are also very different. What you can see here, the timescale is it takes 10 seconds of no assembly, about 8.7 here, and then assembly begins. Here, within a fraction of a second, you can see that there is assembly, a considerable amount of assembly, because the conditions are aggressive. To analyze this data, we had to combine maximum entropy optimization algorithms, which correspond to an optimization method with minimal bias. What it means is that it gives you the broadest distribution of intermediate that the data does not exclude. As you can imagine, from maximum information entropy, it will try to give you the fit with that at the same time has maximum entropy, information entropy. And that's why if the intermediates are not, if the data that cannot exclude those intermediates, they will be included in the distribution, and it will be the broadest and with minimal bias. However, doing just maximum entropy is not enough. We couldn't get physical relevant. We could get a good fit to the data, but it wasn't with physical, it wasn't consistent with other physical information that we had. So in order to analyze these data, we had to do another thing, which is to take prior distribution of intermediate of states that take into account the thermodynamic stability of those intermediates. So by combining maximum entropy with thermodynamic stability, we could filter out the physical distribution that corresponded really to all the information we had about the system. So in mild reaction conditions, these are the data again, and they're analyzed, and as you can see, that now the data are overlaid. You can see very little assembly in the first seconds of assembly. The first second, this is now the mass fraction as a function of number of dimers. So you see the dimers remain in the beginning, the first second, and this is the time axis. And what you see here is also that the curves cross at isospastic points here, or nearly isospastic points, which means that we only have accumulation of either the products or the dimers. And intermediates exist but do not accumulate very much. Here we have the mass fraction displayed as a function of time for the dimer. And you see the lag phase here where no assembly occurs, and then suddenly assembly occurs. T = 4 starts to form. There is a little bit of 2 and 3 dimers that accumulate a little bit. That's the only thing that accumulates. This is very different from the state where the aggressive conditions occur. So we go through a very narrow path here, from dimer all the way to ascend to the full capsid. And this goes through very compacted states, as I'll show you later when we take into account the energy landscape. So here is the aggressive conditions. Here you see that the data does not, there are no asymptotic points anymore. You see the data is very different. The time scales here is the first quarter of a second. And within the first quarter of a second, already particle formed, the full particle formed, the 3 equal 3, much more T equal 3 is forming. And there is a broad distribution of intermediates between 7 and 35 dimers. And as you can see, they appear and disappear. This is the mass fraction of those intermediates. And the dimer immediately start in the first second already to decrease. The particles immediately form. And within a second or so, the first very fraction, 300 milliseconds of the assembly, the pathway for assembly is already determined, and the whole thing is really, really happening very fast. And now, because we had and then we also have the intermediate ionic strength, where we have intermediate, we have just 10 dimers as an intermediate. So decamer would be stable in the energy landscape, and therefore it appears, and things are behaving as an intermediate state. Now, to give rationalize, because we had this energy, all the enumeration of states and the thermodynamic stability and the free energies, we could plot the energy landscape and of the reactions in the 3 cases. So what I'm showing here is the same phase of degree of connectivity. Here is between 0 and 1, where these are the compacted structures, these are the holy structures that many holes have many holes here. And this is as a function of the number of dimers. So if we are in weak assembly conditions, below the limits below the 7 kT that we need for assembly, what we see is that the pathway for assemblies are all blocked. This is the minimum free energy, and all the assembled structures are at much higher energies, and therefore there is an energy barrier for assembly. If we go to mild assembly conditions, which are close to the physiological assembly conditions, what we find is that the energy, as you can see here, these are the minimum free energy is at the capsid and at the compacted structure and close to the beginning at very low number of dimers. And this energy landscape corresponds to a very narrow pathway for assembly. Alternative assemblies are blocked. There is a high barrier to cross here, to go, and assembly is directed towards the compacted structures. So dimers assembled into intermediates that are with very high degree of connectivity. There is also a very high barrier for assembly, which allows this is the leg phase, the origin of the leg phase, because it allows the particles to assemble and disassemble and correct the errors during the assembly. And eventually, well-formed particles can, once they cross the barrier, they will not cross back, and they will fall into and they will continue to the well-formed particles. So this is the conditions close to physiological were good conditions and for successful assembly. When the assemblies have aggressive conditions, the barrier for assembly is very low. The attraction is so strong, and we're talking only about a small difference, but it's on many subunits. And here what we see is that the minimal free energy path is indeed through the compacted structures, most compacted structure. These are the most stable intermediates. However, in this contour, what you can see here is that there is no barrier to cross to other regimes. And for example, this broken black curve is, there is no energy cost to go there. It's not the minimum path, but they could go there, and they do go there because the reaction is very quick and there is no time. And once the particles went there, there's a high barrier of energy to disassemble because the association energy is strong and many subunits has to dissociate. And therefore, the intermediates are kinetically trapped in states that are not stable, the most stable state, but they are stable, they are very, very stable, still very stable. And this is why we are getting these intermediates to see and malform, and eventually even malform particles. That will take time until they can go to the final product. So reversibility is crucial to correct assembly of capsid. This is obtained in the case of mild conditions, where we only go through the very narrow path of assembly. And however, the path gets broader and broader once the assembly conditions become more aggressive and the association energy between dimers is stronger. Malformed particles and go through intermediates. The system goes through a broad distribution of intermediates with many holes that are stable enough and are kinetically trapped. Okay, so this is, to summarize, we were able to use SAXS and time result SAXS to detect the structures, the interactions, and the assembly pathways of in native conditions of HBV, hepatitis B capsids. We see that we were able to vary the condition between successful assembly, where it goes through, the assembly goes through a narrow pathway. However, off-path assemblies occur when the assembly conditions become more aggressive, and the only difference is 1 kT. So the geometry of the particles describes the possible states of intermediates, but energy is tunable by ionic strength and temperature, but the energy is making the actual selection of which of the assembly pathway, which intermediates will be available, will be taken eventually. And this is how the system solves itself. So just one word about disassembly of a different virus, just to mention it if there is still time. So in the case of SV40, it's a different virus with the genome. It's an icosahedral virus with it's a slightly larger diameter, 48 nanometers. And these are cryo-EM images. We studied here the disassembly using time-resolved experiments. And we found that in this case, where the DNA is encapsulated and condensed by histone proteins, nucleosomes actually, then once the pH is becoming above 10, then the nucleosomes become swell, and the capsid swells. So there is a lot of pressure inside. Eventually, there's a hole that is being poked, and the nucleosomes is ejected completely. And then eventually, there is disintegration of all the subunits. So we were able to see it also in cryo-EM and also describe the mass fraction of all of these 5 states, 5 representative states, as a function of time in this process. So this is the other side of assembly, is the opposite is disassembly. And finally, I should thank Roy Assor. He's the PhD student, he's now in Oxford University, that did all of this work, the rest of the group. And this is a collaboration with the group of Adam Zlotnick, and it was done at the SRF, the IT2, and funded. These are the funding agencies. And thank you for your attention.